Thank you for braving the weather. So I can't even say New Yorkers are tough because I know you're from all over the tri-state, so we're all tough. But thank you for making it in. Welcome. Good morning, President Lynch, Dean of Research, Dr. Wang, President of MacCube, Dr. Nolan, and the rest of the MacCube Executive Committee. Thank you for giving us the chance to host this conference. It is an absolute pleasure. As I was telling our president, it's almost 10 years in the making. We've been trying to host this conference for a very long time, so I'm so glad that you gave us the chance to do that. We're so happy to see everyone here on our campus. So what does MacCube mean to me personally as a faculty? I've been attending last 12 years straight. This is the 51st conference. I am Nidhi Gadura, by the way, chair of biology here at Queensborough. So MacCube membership represents 72 campuses, and a whole bunch of our students, I know, will end up at these senior colleges and whatnot, so it really means a lot. And today we have almost 400 people who are going to register and be part of this conference. We have over 140 poster presentations. So give a big round of applause to all the students. There's 17 faculty member presentations and 2 hands-on workshops. So in your programs, it says Medical Arts Building. Note it's in Science Building. Both buildings are connected on 2 floors. You're sitting in Medical Arts, but right next door is Science Building. So it's pretty much very next to each other. But faculty presentations are in Science Building. So we have a great list of sponsors with us today. So make sure that you guys visit their booths. We have senior colleges who are here to recruit really good students. We have a whole bunch of publishing companies and instrument companies. So for all the faculty, make sure that you're visiting them, please. It takes a village to put a conference this size together, as you can imagine. And I have almost, I would say, good 75% of my biology department here at Queensborough who's involved. So I really want to appreciate and thank all the faculty and CLTs who are helping us running around for months, contributing their time and energy. Special thank you to the chairs of this conference. So chair and co-chair, Dr. Sullivan, Dr. Mader, say hello to everyone. They put in a lot of hard work. Co-chairs of posters, making sure you have poster numbers, making sure you guys are going to get judged, making sure that the scores are tallied, are Drs. Golubiewska and Dr. Javdan. So a round of applause for them. And member presentation chairs, Dr. Negwin and Dr. Tade. So they're going to make sure that members are all taken care of as well. Big thank you to dedicated buildings and grounds, so B&G, really we appreciate all the work. They've been at it since yesterday, putting all the tables and chairs. And all of you who are done putting their posters, it looks beautiful, doesn't it? Yeah, thank you. Our Academic Computing Center, our marketing department, media services, thank you so much for all the help. I truly appreciate the support of our administration. We would be nowhere without that. So this conversation started with our previous president, Dr. Call, and Dr. Lynch, who was now our president, absolutely unwavering support that they had offered for this conference to happen. So I truly appreciate that. And that support means a lot to us as faculty members. We cannot pay you back, but we always pay it forward to the students. So we totally appreciate that. And without further ado, I would like to welcome President Lynch to the podium, please, to say a few words. He's the man who paid for breakfast.
Well, thank you, Dr. Gadora. I promise to keep my remarks brief, but in full disclosure, that's because the prepared remarks that I had intended to deliver when I hit print this morning, I printed out my daughter's college application essay. So in my pre-coffee Saturday morning stupor, I don't think you want to hear about her plans to pursue a degree in history, much like her father. I think her mother, who's a biologist, would much prefer that she follow in her own footsteps. But so I will give some very brief remarks. Again, I wanted to echo Dr. Godora's grateful remarks concerning the contributions of members of the Department of Biological Sciences and Geology in putting this conference together, as well as the hard work of our facilities teams, particularly public safety, buildings and grounds, who've made the campus look so beautiful for today's events. This campus, as I'm sure you'll notice, is one of the more beautiful within the City University of New York. But it's not just the aesthetics that make it so appealing. We're located on a former golf course, so there's a lot of open green spaces and rolling hills. But what really makes this campus so beautiful is the commitment of our faculty to supporting student success initiatives. When I took this position 2 years ago, I was amazed at the level of activity that was happening in our classroom involving our students. Real hands-on applied learning experiences where faculty members were involving students in undergraduate research activities, getting them involved in meaningful research that resulted in scholarly presentations and publications in journals of some renown. And I thought this is exactly what we should be doing at a community college. It's exactly what we should be doing as a campus of the City University of New York, providing opportunities for our students, many of whom are first-generation students, to pursue higher education in a way that many of them would have not known was possible. So today's workshops, presentations, poster sessions, are a reflection of the mission of City University of New York, of QCC, and of MACUB in providing gateway and transformational educational opportunities for our students. And I wanted to, again, acknowledge the role of our faculty in foregrounding student research in the classroom and beyond. QCC is one of the most diverse campuses in the country. We're located in the most diverse county in the nation, and that diversity is reflected in our faculty and staff and in our student population. It's also reflected in the variety of our academic programs. We offer more than 40 academic programs. We have an 80% transfer rate to our four-year institutions. And as you'll see by some of the presentations that our students are involved in today, we have a diversity of interests when it applies to our academic programs and our academic efforts. At this time, I would like to gracefully exit the stage and allow the president of MACUB to come up and make some additional remarks. So again, welcome to Queensborough. Have a fantastic conference. I hope to see several of you at the poster sessions, and welcome again. Thanks.
Thank you, President, for those wonderful remarks. And thank you, Needy, and your team for putting on this great conference. And give yourselves one more big round of applause for coming out in this weather, this nor'easter. And I'm just going to acknowledge three people from our board. Gary Sarinsky, stand up. Okay, and we have, I forgot to mention Paul Russo. He's over at the desk. He's still registering people. So make sure that you pat him on the back or bring him some lunch or something, because he won't even take a break for lunch. So give a hand for Paul, even though he's not here. And I want Ed Catapan to stand up. Thank you. He is the editor of In Vivo, and it's a peer-reviewed journal, so faculty members that would like to publish something, send it to Ed. And then Margaret. Carol, where's Margaret? She's our treasurer. And you can see the other names of the board members who are very helpful that are in the program. And somebody asked me last night, how did I meet Greg Dunn? Because I chose the two speakers for this year's conference. And I actually met him through the second speaker, who is Allison Dell. And Allison Dell is a professor at St. Francis College. I'm the chair of biology and health promotion and healthcare management at St. Francis. So not to try to take you guys away from CUNY, but if you want to transfer to St. Francis, we'd love to have you. But anyways, we took a group of students, and it was probably our third trip, to the Mutter Museum in Philadelphia. How many of you have been there? The Mutter Museum. It's a wonderful museum. You can get there on Megabus for like $20 or something. And so $40 round trip. We charge the students. They got to make their own little bus trip and bus fare. And it's a wonderful museum, and it has like wax models of different diseases. I mean, if you like gross things, as biologists, we're kind of all into that, right? And they have skulls and skeletons, and it's a very exciting museum. And they have a separate art gallery off to the side. And so it's a temporary exhibit. So I went through the exhibit, and I was with Dr. Dell, and I said, these paintings are really cool. They're of like these neurons. And one of my favorites was of like, it was a city, but they turned it into neural circuits. And I said, wow, this artist, like really fantastic. And so then Allison Dell says to me, why don't we try to get him to talk at St. Francis College? So we have this program that Allison and Arena Ellison from Mercy College, she's at Mercy College now, she was at St. Francis for a time, started this Art in the Lab program. And so we invited Greg Dunn to come and speak and to tell us how he does his artwork with neurons. So, and he hails from the University of Pennsylvania, and Ph.D. in neuroscience, and he is a neuroscientist / artist. So you can read a little bit more about his bio and the program, and check out his website. It's absolutely wonderful. So without further ado, I'd like to welcome Greg Dunn.
Thanks, Kathy, for the nice introduction. Great to be here. Thanks for packing the room. It's awesome to have people standing in the back. Although I pity you, I'm glad that you're here. Right, so I guess I wanted to open my talk just by giving what kind of how I think about the spectrum of art and science, because my definition might be a little bit different from what you're used to thinking about, but I think it's important to start there. So I wanted to present to you a painting by Alyssa Monks, which is an oil painting, incredibly. I mean, the technical skill to be able to pull this off is formidable. And yet, I think that this is a science experiment, not a work of art. And my rationale is that if you had 100 very technically gifted artists, you set them up in this room and asked them to paint this scene that was in front of all their eyes, that the results of their experiments would be identical. You know, controlling for perspective, of course. That's what science is trying to do, right? We're trying to perform experiments, learn something about our external reality so that we can agree that something is true or not. Whereas on the other side of the spectrum, this Gerhard Richter canvas, which is an abstract painting, if you were to give those same 100 artists a couple tubes of paint and to say, I want you to paint frustration or anger or joy, there's a degree of introspection that needs to occur because there's no defined external source of inspiration for that. We can all agree roughly on the bounds of what that feels like, but the results of those 100 experiments would be different from one another. Art is the process of introspecting and bringing a subjective experience into objective reality for others to be able to appreciate. And by that definition, almost everything that we do is a combination of art and science. Scientific art, in my opinion, is not so much about what it is that you're choosing to paint. Am I, for example, choosing to paint a neuron because scientists are studying it at this moment in time, and does that make it, in and of itself, science art? Or am I using the scientific method in my artistic practice and integrating it on a conceptual level? So this piece by Craig Mullins, this environment doesn't exist, but he understands very clearly the rules of perspective and light and reflectivity and is able to integrate our scientific understanding of how those things work and create a new environment for us to be able to appreciate. And in the realm of scientific imagery, what would be kind of on the most hard, verbatim, scientific type of images you can get is, for example, a photomicrograph. This is just a slice of tissue stained with fluorescent proteins. Many of you have seen an image like this. And then you can paint the brain much more kind of abstractly, right? So this is going to define the artistic side of this. And I'm going to go ahead and make the statement that Ramon y Cajal, the revered Spanish neuroscientist who won the Nobel Prize in 1906 for his work in underscoring synaptic theory and his work with anatomy. These are his drawings right here. And what you can see is that there's a photomicrograph on the left of the set and his drawing of that on the right. So right now, with today's technology, you can take a slice of tissue, a three-dimensional slice of tissue, you can take thin sections through it with the microscope, and you can more easily create a three-dimensional understanding of what it is you're trying to look at here. That was not the case in his time. He's methodically taking sections, he's staining each one, he's looking at each one independently from one another. And he could have been tracing what he was looking at using the camera lucida technique he had, where it's a kind of a projection of what he's seeing through the microscope. But he wasn't doing that for the most part. He was looking at all the slices, he was integrating what he was seeing using his scientific knowledge, and he was using his artistic skill to kind of condense it into something which is actually more readable than would be if you were to give one of these slides to any one of us in the audience and have us kind of decipher what there is. I think it's important that when you're doing this, that you're clear about how you're kind of blurring those boundaries so that others can kind of know what that means. But I'm also making the statement that including art in the scientific process can actually more forcefully and clearly present your ideas than just a verbatim kind of scientific presentation. And as an artist who's concerned with the brain, I'm often asking myself, how does the average person just walking down the street really think about their own brain? Like, how do you conceive of what a lot of people think is the most complex object in the known universe, which is at the core of our consciousness and being? And this is kind of the depressing answer. This is the type of image that you would see in Scientific American and the New York Times. This is kind of the meme of the day, this blue outline of the brain with lightning bolts coming out of it to emphasize its magic. This is a common trope into how to depict neurons as well. And what happens, and this is not to pick on illustrators at all, this happens in every field. You have an incremental kind of step forward in the visualization of something. And when you're talking about images like this, since we think visually for the most part, when you have a step forward in how to depict something, oftentimes other people will kind of imitate that, or they will be inspired by it, and you get a large body of work that looks like this. And to be fair, this is trying to communicate a relatively straightforward point, which is that neurons, as you see here, are sending little electrical potentials down their axons, which are being collected by their dendrites. Basically, that neurons are firing these little bolts of electricity to one another, and that's the basis of communication. So this tells you something very fundamental about the brain, but it doesn't come close to showing you how damn complex it is. You start to get into more of the scientific imagery here. These are fMRI images, functional magnetic resonance imagery, which is done in an MRI scanner, and it's looking at the oxidation state of iron in measuring blood flow, which is kind of an indirect measure of brain activity. And here, this might be what it looks like when you feel like your butt is itchy. And it's telling you that there's something compartmentalized about our brains, right? Like, that's the information that's being transmitted here. It's not telling you really much about the anatomy, particularly not on the microscopic scale, but it's telling you that we use different parts of our brain for different things. And tragically, although I'm not positive about this, I suspect that these types of images gave rise to the popular misconception that we only use 10% of our brains at a given time, which is complete nonsense. And if that were the case, our brains would have been evolved out of our bodies almost instantaneously for such an energy-hungry organ. It would not have flown. So now we're looking at photomicrographs of small slices of tissue. Now you can see, all right, well, there's clearly a very distinctive and complicated kind of two-dimensional anatomy that I can see here. This isn't telling you anything about what's happening in 3 dimensions, not telling you anything about what's happening in time, and these microscopic images tend to be confined to a small region that you can fit in the field of view of the microscope. And of course, we all know what this looks like. This is the brain as it comes out of somebody's head. And I remember dissecting a brain for the first time in grad school and scratching my head a little bit and being like, this doesn't, like, where's the little, like oval shape that's supposed to be the nucleus accumbens, for example, that you see in your textbooks, and it's hard to decipher really what's going on here for the most part. You can see the white matter, you can see the dark matter, or the gray matter, and how they're interacting, but for the most part, this does not even come close to belying the beautiful complexity of this universe. So when I began my artistic explorations of the brain, it was through the scaffold of my love of Asian art. This is a piece, this is a folding screen from the Edo period in Japan. And one thing that I really love about this artwork is their emphasis on the spontaneity of line and how that kind of random branching pattern that they love to explore is being put on this gold background. Gold's an excellent medium for this because you get this kind of subtle reflectivity coming off the gold, which really helps to cast the delicacy of the branches in this case into a really kind of nice silhouette. And that's really what this art is about. It's about negative space and it's about silhouette. And sure enough, the silhouettes of neurons look very, very similar to what you would see, in a tree or branch, in our kind of macroscopic world. And the point that I'm trying to make in some of my early work is that our universe is organized into fractals. And a fractal is a shape which repeats at many different orders of magnitude. So you can look through a microscope and you see a neuron, you see a shape like this. You can see a lightning bolt or a crack in the pavement. But you can look at the orientations of galactic superclusters. You look at some of these images, which is showing how the galaxies are arranged, and sure enough, they look like neurons too. This shape is just simply woven into the fabric of our universe. So these were the kind of artistic experiments that I began to do. This is a piece called Basket and Pyramidals, where I'm putting the argument in the familiar context of Asian art. I think that's oftentimes an effective way to introduce an idea, give some ground for people to stand on, and then push them a little bit. And upon further analysis, even somebody who doesn't have a neuroscience background would be like, it's not really a tree, but it kind of looks like it. Wait, these are neurons? That's what neurons look like? That's kind of the idea to broaden somebody's horizon a little bit with an image like this. And these are further kind of experiments along these lines, couching it in the Asian aesthetic. These are hanging scrolls. You have an image of the retina on the left, of astrocytes in the middle, and of kind of conceptual piece featuring pyramidal cells on the right there. And I began to get a little restless and start kind of wanting to experiment more with some aesthetic techniques. This is a piece done in 2012, it looks like, which is a process I call deep reflective gold leaf. And you can see here this kind of bright region, these bright regions right here, are not due to the pigment on the canvas as much as it's due to light being reflected off the canvas. The pieces are not literally iridescent in that they have photonic crystals on a nanoscale which are splitting light, but there's a differential etch in the surfaces of these metals which is causing the painting to kind of become alive as you walk around it. So the reason this is bright is that there's a light above it right here which is shining light down like this, and the background is etched in a horizontal way right here. So you can see these neurons right here. These guys are the layer 3 pyramidal neurons of this section of cortex. And you can see when they are on top of the background, which is brightly illuminated, that they're cast into silhouette. So you're playing with contrast here. That's what your brain responds most to, is when you have contrast between things. Even though these two objects are brightly reflective, one is more than the other, so your brain will make one appear as dark and the other appear as bright. So these are kind of the the origins of some of the experiments that I'm going to talk about at greater length today. This is excuse me, another piece along these lines. This is called Spinal Cord. Again, kind of drawing parallels between the anatomies of a butterfly and the spinal cord cross-section here. This is a piece called Deciphering Spikes. This is a 64-unit electrode which is being inserted into layers of cortex and is kind of reading the brain activity here. And this piece is really about the very bizarre concept that we evolved from primordial ooze. And over billions of years, a human brain evolved. And one of the things it wants to do is to figure out what it is. So this is about kind of that recursive loop. And it's styled as almost an alien abduction to make that point. This is a piece called Axon Pathfinding, which is about the chaotic early developmental stages when you have many, many axons growing towards their targets and interacting in beautiful ways. There's a piece called Hippocampal Formations, which is as much about the hippocampus, which is the part of our brain that controls learning and memory and episodic experiences, integrating sensory experience. So it's half inspired by that and half by erosion patterns seen by satellites from space. In the Jordanian desert, you have, this would be the mountain range here, right here, and then water is shedding off in these kind of rivulets and forming in these kind of canals here, which are shedding water off the eroded area. So it just, again, calling into question this idea that nature is following the path of least resistance, and because of that, you get similar imagery across different scales. So it was about this time when I was in grad school, I became obsessed with the idea that I needed to show something more than this. Because one of the most humbling things about being a neuroscientist is that the second you think you understand something is the second you search the literature and find 6,000 papers that have looked at either exactly that thing or things kind of within that general vicinity, and you're humbled by how little you know. And then you consider the fact that we know a fraction of a % of what's actually happening in the brain. So to be able to communicate the beauty of that complexity, I mean, obviously, what's going on in here at every moment and right now in every one of your brains has to be incredibly sophisticated to give rise to this experience. So that was why I embarked on this project called Self-Reflected, which was funded by the National Science Foundation, and this was done out of a couple years of work at the University of Pennsylvania. Physical object hanging on the wall. This is not a computer simulation. This is the collective activity of 500,000 neurons at once, kind of encompassing half a microsecond of brain time in a repeated loop. This is the most complex artistic depiction of the brain in the world. This technique is done by a process called reflective microetching, which is the microscopic control of how a metallic surface reflects light. And one of the cool things about it is that color has been turned into an infinite variable here. It's merely reflecting the light that you're putting onto it. So you can emphasize things with certain colors, you can animate using other colors. This is a demonstration where a board is being passed across the lamps which are illuminating the piece, which is showing you the kind of the channel specificity of how a microetching works. So I'll give you a little bit of a tour of the brain now. We're going to start, so we're at a sagittal slice, which is in this direction. We're going to start at the very back here. This is the Parietal Cortex. And this is a type of calculation that's happening when you're reaching out to pet your cat or to pick up your bottle of water and take a sip, integrating kind of vision and movement. We're getting in here now to Somatosensory Cortex. Many of you know the famous homunculus. This is what this looks like. So this is how kind of you're running your hand over your skin. These are the types of calculations that you might see. And you can see this very specific kind of layered structure to the cortex here. This is the outer covering of the brain. Here's the motor cortex. This has some of the largest neurons in the body, the layer 5 BET cells, which are firing motor information into what's going to become the spinal cord. Here's premotor cortex, which is involved with similar types of actions. Now we're about right here. And we're getting into the frontal cortex now. And what you can see about this region is that the neural structures are quite different here. There's not as much of that distinct layered aspect to this region. There's a lot more inner neurons. The neurons are more complicated here than they are in the more ancient sensory regions. this is the part of the brain which separates us from even the higher primates. This is what logic and planning look like. And interestingly here, at least in this slice that we're in, information is flowing unidirectionally from the frontal cortex into the basal ganglia and into the kind of more primitive emotional centers of the brain, the limbic system. This is kind of the correlate of the top-down control, you know, how we use our kind of conception of self and logic to be able to kind of control our baser instincts. That's sort of what's happening here. And now we're getting into the basal forebrain and the nucleus accumbens. So this big bright flash right here would be what it looked like when you snort cocaine or do other pleasurable things. This is the basal ganglia here, the caudate nucleus. This is integrating all sorts of things like motor information, sensory information, memory information to help us plan how we're going to move. Now we get into the thalamus here, which is kind of the central structure of the brain, where tons of stuff is going on here. So much complicated activity, so many recurrent connections are coming through here, and it's sort of filtering them and deciding what to bring into our conscious experience. Here's the VTA, dopaminergic nucleus here. There's the raphe. There's some other important nuclei up here in the midbrain. This is becoming the spinal cord right now. So we're in the brainstem here, and this contains very basic kind of functions for life, like breathing and hunger, that sort of thing. Now we're getting into the cerebellum, which is right in the back of the brain right here. Cerebellum meaning little brain. This region contains half the neurons in the whole body, and it's calculating where your body is in space and also calculating some movement information. I would say that in order to actually depict what the anatomy here looks like, this piece would have had to be probably 100 times the size that it is. It's already the size of a wall to pack all that anatomy in. You have the inferior and superior colliculi here, which are for hearing and vision, respectively. The lateral geniculate nucleus, which is receiving information from the retina, is there on the left. And then it's sending its projections in this wave-like fashion into this kind of mouth of neurons here in the calcerine cortex, which is called the primary visual cortex. Very basic calculations of where the angles are in your visual field, etc., are happening in this region of the brain. And then that information gets sent to higher order visual cortex. So that's that piece. It was done in collaboration with Dr. Brian Edwards, who's an applied physicist at Penn, who could not be here today, but who is an effing hero. All right, so how does a microetching work is a logical question to ask. Basically what it's doing is that it is separating objects in reflective space. It's adding a third dimension to a two-dimensional piece of art. So this is a close-up of the surface of microetching. These little etches are generally too small for your eye to see, but you can see what appears as red is going at a different angle than what is appearing as green. There are lights above the piece here, which are different colors, and if you have a perpendicular relationship between the photons arriving to that etch, then that etched surface will appear as that color. This also means that there's no fixed color anywhere in the piece, and that as you walk around it like this, that all these colors would be shifting. And the one maybe pain in the ass about these is that when there's a lot of people standing there looking at them, I can't point to something and expect that you would be seeing the same thing, because we're all seeing different things on the surface here. So this is what the surface looks like under a scanning electron microscope. The image on the right is 1200x and on the left 120x. So it doesn't look really much like anything when you're looking at it very close up. It's only when you kind of zoom out that you start to see these more macroscopic changes. All right, so here, this is an example of a data set that we use to actually do the etching. And what I want to call your attention to is this little curlicue right here, which is a dendrite for this neuron. You can see how the etch angle kind of changes as you go over this. And what that means is that when you strafe a light source over the top, that you're going to get a bright spot here initially, only at the tip here where it's optimized toward the light being here. And then as the light moves like this, you'll see that little spark of light sort of move along the structure until it gets to the soma here. So you can actually animate things using this technique. And that's the basis of how all that neural activity was animated in the piece. So as I mentioned, this project was funded by the National Science Foundation after we had an exhibit of some of our early microetchings in their headquarters. And we are forever grateful to them for believing in this project. And one of the first things we had to figure out was, what are you going to show? Because there's so much cool stuff to show. And we eventually decided on a sagittal slice of the brain, which I mentioned is in this direction, because it's the only non-symmetrical slice. Like an axial and a coronal slice are both symmetrical, so you'd be showing relatively redundant structures. And we ended up tilting the slice a little bit here so that we could avoid the ventricles, these fluid-filled cavities in the brain, which would just show up as holes. And because there's a lot of circuitry that's kind of interesting along this plane, so that's where we decided. This was the state, so we started this project in 2014. This was the state of the best human data set at the time in terms of overall anatomy. And you can see there's little stripes going through it because they actually slice this in this direction, not in this direction. So this is a kind of a reconstruction of the image. Basically, the point I'm trying to make is that the data sets were crappy at the time. And we had to mine really, really deep in order to get the quality of information that we needed to find. Because you can't just zoom into this image and see what the different neural cell types look like. You've got to kind of build it from scratch. And one thing I really want to emphasize here, too, is that there were a lot of unknowns in this project. There's a lot of things that could be looked up, there's a lot of things that can be inferred, but much like I was kind of getting to with this Cajal point that I was making in the early stages, is that there was also kind of filling in the blanks logically to try to make a point more forcefully, like a structure being slightly moved into or out of the plane of the painting in order to show a circuit in its entirety. Right, so this is what all the different brain regions in our slice looked like. Each different color indicates a different kind of functional region of the brain. There's more than this, but we called it a day here. And for each one of these regions, there were three neuroscience students who were helping us with this project, who went through for each of these regions, combed through the primary literature, and dug up a whole bunch of information about what the neurons look like, what they're connected to, what their firing patterns are like, what their, the functional consequence of the region is, blah, 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 blah. And we assembled a whole bunch of data in that regard to kind of, to inform the creation of the piece. And then, let's see if I can turn this down. This is kind of loud. To create the neurons themselves, we painted them all by hand, or by breath, actually. And the reason that we use this technique rather than scans or rather than painting by hand is that when you try to paint a random branching structure with your hand, you quickly realize that your brain sucks at that. It's always wanting to make kind of regular patterns out of things. And you discover it's making a regular pattern and you say stop it and you try not to do that, and then some other regular pattern comes out. Your brain's not really good at generating randomness. Nature's really good at generating randomness, and it wants to do it all the time. So that was the creation of this technique, was to use the turbulence of the air coming through this straw, essentially, as creating this kind of stochasticity to it. You practice the technique, you get better at it, but at its heart, there's always that element of randomness to it, which I think is more reminiscent of what the neurons actually look like anyway. So we painted about 5 versions of each type of neuron, and there were maybe 100 to 150 or so different types of neurons in the piece. And then these were scanned into the computer and turned into vector objects, which is essentially a mathematical description of the outline of each one of the neurons. And once you have it in math, then you can change it as much as you want. You make it small, big, rotate it, stretch it, whatever you want, without loss of resolution. So using some vector art programs, we painted down about 1 million or so of these neurons across the entire brain. This is just a small section of cortex. There's maybe a couple thousand neurons here. So that's what the creation of the gray matter looked like. But as we know, the brain is only half gray matter. The other half is white matter. These kind of highways of interacting axons and traces which are coursing through the brain. This image on the right, anything which is darkly stained, is against the myelin sheath of axons. So all this stuff in black here are white matter tracts. And they're all kind of twisting around one another in these just gorgeous patterns that is impossible to decipher using an image like this. And that's why we turned to some buddies at the Carnegie Mellon University. This is my friend John Piles going in the scanner. And we did some diffusion spectrum imaging of the brain. So this is of a sagittal section. This is the basically, it uses a powerful magnet to be able to detect the direction of diffusion of water molecules in small voxels, which is a three-dimensional pixel, and then reconstruct where all of these tiny little tubes are going in your brain. This is the data set filtered for our plane of interest, again, like this, coming through the brain. And this was used to inspire the creation of our data sets that we used in the final piece. This is not used verbatim, but it sure is pretty. So using that, we were then able to make these types of sets. These are kind of like vector line bundles. These are both kind of areas of the cortical sheet that we ended up using. So many files were made in that regard, and now what we have at this point in the project is a black and white image of a very complex kind of array of neurons and a very complex array of axons, and that's already like it was a formidable point to get to here. This was maybe a year and a half worth of work or so to get to this point. But what happens next is really what brought the project to life and is what brought that extra kind of dimension of movement to the piece, the animation of how the neurons are connecting with one another. So if you think about a neural circuit, it's pretty basic, at its kind of root level. You have one neuron which is connecting to another neuron through an axon, sending its action potential. That kind of input gets considered by the receiving neuron, and it decides based on a few variables whether or not it's going to fire its own action potential. So you have kind of step A, step B, step C. This is how those steps were sequenced in the final piece. So this is, we built an algorithm to help us to kind of sequence all this information. So one line of this algorithm is telling you where you're starting from here, where you're going, what kind of axon set you're going to, and then what your destination is here. And then all these different variables are controlling things like speed and kind of fidelity of connections, whether or not they're firing as a burst, et cetera. So lest anybody think that this was, a bunch of scatterbrain artists like going into their studio and just being like, what is going to strike me today? This is much more like a bunch of burnt out hippies being forced to do like brutal taxes all day. Right, so let's look at a sample scenario here where we have this guy, this neuron right here, which has connections from 3 neurons, here, here, and here. And let's say in order for this neuron to fire, it has to receive input at the same time from these other neurons. This essentially turns out to be a time problem when you have different distances between these neurons. Assuming that their action potentials travel at the same rate, this one is going to have to fire first, and by the time its action potential reaches here, then these two can fire and they'd all kind of converge at the same time. And what this means is that when you're working with these kind of mathematical problems, that things get pushed around in time in order to satisfy the constraints. So what you're going to see in future images here is that time is going to be represented by color. You can think of a microetching as being kind of like an animated GIF. There's maybe 150 to 200 frames in a microetching, and we're going to call a frame at the very beginning of the microetching red, and then later ones are going to be orange and yellow and green and blue, etc. And what that means is if I'm actually standing in front of the microetching itself, let's see, that's the microetching, I would be seeing as bright all the red pixels here, then I'd be seeing the orange ones and then the yellow ones and then the green ones. In the microetching, they're not literally those colors. they would just be brightly illuminated as you're walking across it. So I'm going to show you some color maps now of how we program the time. So you can get a more of a sense of what this is going to look like in time. If you're going to animate this, you would see the red pixels and then the yellow ones, or the orange ones, yellow ones, blah, blah, blah. So you can see that this sort of densely packed region of layer 5 motor cortex here is going to fire relatively late in the animation because blue is late in the spectrum. This is what the data set looked like for the entire brain prior to etching. This is about a week's worth of time rendering for the computer that we had, which is a fast computer. The math here looks similar to if you're going to deliver 10,000 packages via FedEx and you're trying to optimize the most efficient route to save gas. That's generally the category of what we're looking at here. So let's zoom in on a small part. We see there's maybe, 2,000 pixels in this image. And how are we going to translate this data into that physical object that I keep telling you about? We have to make another algorithm to solve that problem. So let's assume that the white square is going to be the microetching. The very first thing you need to know is where is the light going to be? The light is the absolute heart of a microetching. A microetching doesn't look like anything without light. So you need to know where that is first. Let's just choose a pixel in the middle of the etching here. So you know what the angle is going to be between that point and the light source. And based upon the color of that pixel that I just showed you in the previous slides, we also know where we want the viewer to be able to see that pixel. So we can then draw a vector in that direction as well. So now we have two known angles, which means that we can then calculate the angle at which you would need to make a tiny little reflective mirror at that pixel in order to get the light to exactly where you want it to go. In reality, however, pixels are square, not linear like this. And I should also say the etches are cylinders because it distributes the light in such a way where you can see it if you're short or if you're tall. Unlike if you had a beveled surface like a diamond, you'd get a brighter reflection but focused on a much more specific spot. Right, so we're making little square fields of these reflectors. And this is what that data looks like after this step. So we've taken essentially a 300 dpi image, which you're normally used to dealing with, printing on your printer, et cetera, and blowing it up into a 5,000 dpi image in order to cram all this information in here and to be able to get the degree of angular specificity that we need to get in order to do these experiments. So this is way subretinal. You could not see this printed onto a transparency. This would be viewable under a microscope. The distance between these lines is about 30 to 50 microns. These are the transparencies that were printed in order to do the following steps. This is just hanging up in the clean room in my studio. So you take one of these transparencies and you put it down onto a photoresist. So let's say this down here, the substrate's just a sheet of steel. This is a laminated film of photoresist. Photoresist is a polymer surface which is sensitive to ultraviolet light. It's oftentimes used in the creation of silicon microchips and fabrication of microelectronics. So the transparency is on top here, and you can see here's an area with black printing, here's a clear area, and here's an area with black printing. This red here is ultraviolet light that you shine on the entire apparatus. So it penetrates wherever it's clear, but the black printing acts as a resist, and it keeps it protected from that ultraviolet light. And what happens is you get this kind of polymerized pellet at this spot. It makes it hard wherever it's able to burn that resist, wherever it's not protected. You turn off the light, remove the resist, dip the entire plate into a weak basic solution which eats away the soft parts. So anything that was underneath that black printing now has become a little divot in the surface of the etching. And this is the lamination of the photoresist in the clean room with Becca. This is what the exposure unit looks like in the studio. This guy right here is the plate being exposed in the clean room, and this is the light source that's spraying that UV light onto it. And this is somebody slacking off on their phone on the right. Probably me. And then we apply gold leaf to the surface. This purple surface that you see here is the color of the resist as it comes out of the previous steps. You can see the reflectivity to it here, but it's not a terribly reflective surface. And because his artwork is essentially all reflective, we want to increase the brightness of that as much as possible. And gold ends up being the perfect solution to this problem because, first of all, it's awesome and everybody loves it. Second of all, it's soft, so you can cram it into all those little kind of nooks and crannies in that fine etching that you've made. It does not tarnish, and it's so thin. It's 200 nanometers thick. By comparison, our etches are 50 to 100 microns wide. That's almost 1,000 times thinner than that geometry, meaning that you could put 50 layers of gold onto it if you wanted to, and it still wouldn't be thick enough to affect that precise topography that you've made. So yeah, putting on a lot of gold leaf. There's like 2,000 squares of it on this. This is a preliminary assembly of the plates. This piece had to be made in 25 individual plates due to the kind of dimensions constraints of some of our of some of our materials. And then they were cut down to size on the metal shears. This is Brian here. He came up with a great method to be able to cut these down to within about 1,000th of an inch of one another, 100th or 1,000th, because we wanted to put them together to have kind of a seamless butting up of one of them. This is an assembly of 9 plates that was done. This is sort of the first day that we realized that it was going to work. And of course, Brian is here worrying about something. Like every good engineer, he's never happy or satisfied. But it looked pretty good to me. These are some of the students in the lab who helped build the light bar. There's a programmable light array that's above the piece in the museum in the Franklin Institute where it's hanging permanently. That was illuminating the piece. And it's basically sending like a little bolt of light down these LEDs. Yes, so this is what the piece looks like under just a single white light. But remember, you can put any color of light that you want onto it. This is maybe 3 or 4 different colors on there. These are some close-ups of the basal ganglia and the thalamus. Here's the cerebellum, one of my favorite regions. This is the parietal cortex. And again, remember what's cool about this too is that the color signifies something. The color here means that whatever is red, for example, or blue, is firing at that moment of time. It's also telling you what the circuit kind of orientations look like here. This is the somatosensory and the motor cortices. Again, zoomed out with the cerebellum and the brainstem. And this is what the piece looks like in the space. It's 8 feet by 12 feet on the wall. Believe you me, there were many times in this project which this was the hardest thing that I've ever made in my life by far, including any experiment that I've ever done in the lab. And there were many times when I was asking myself why we were doing this, because we had to invent a lot of this stuff. You know, there were a lot of roadblocks. And the thing that kept me going was imagining a little kid coming into the space, into this museum, and standing in front of this huge glittering gold work of art that takes up pretty much their entire peripheral vision. And for the light source to strafe across it for the first time, and for that kid just for 1 second to say, wow. And for every one of you professional neuroscientists, for you also, you know, it's meant to be a recalibrator. I don't care at all. I don't care at all if you don't remember that the thalamus has recurrent connections to the motor cortex, or that the cerebellum has, you know, a layer of Purkinje cells that are sending information to the dentate nucleus. All that information is there, and there is a ton of it. This is a highly accurate atlas, essentially. But its most important characteristic is that it shows you through your emotions and through your perceptions what complexity actually looks like. Because I can sit here and scream at all of you, and you've all been screamed at enough, I'm sure, that the brain has 86 billion neurons and tens, if not hundreds of trillions of connections. And it means pretty much nothing because we have no basis for comparison for that kind of thing. If you see what 500,000 neurons looks like, then you can start to say, all right, you know, I'm getting this a little bit more now. And then, of course, I'm going to overwhelm you by telling you that the brain is hundreds and hundreds of million times more complex than this. For example, there's no glia at all in this piece, just to give you a sense of what's missing from it. So that to me is the most important part about this piece, and it's designed to be showing your brain what's happening at the moment it's observing the piece, which is why it's called self-reflected. I think a lot about why art is important and what it can communicate and why it has a leg up in some ways to kind of the dry presentation of scientific facts and why I moved into this field. And one of the most important things that I learned in grad school was how the visual system works, some element of it. Vision is really complex, and there's a lot of brain real estate which is dedicated toward its processing. And it tends to be broken up into parallel processing units, which is a faster method to do it. You need more circuitry, but you can cram through a perception faster than if you had to process this, and then this, and then this, and then this. So information comes in from the retina into the LGN, lateral genetic nucleus, into the cortex, at which point it's broken up into distinct streams. And as a very powerful evolutionary advantage, a stream of primitive form information, I'm talking about just basic silhouette information, is sent to the limbic system before you're even seeing it, before you're like registering what you're looking at. This information is being sent to the limbic system for emotional context. So your instinctual kind of understanding of the world, your emotional baggage, if you will, is being encoded within this visual stream of information, and then is brought back into the visual system and is used to create this perception of what you're seeing. And I'm not talking about there being an emotion associated with the vision, which is somewhat the case. I'm talking about the actual visual information being transformed into you seeing what's not there. This has profound implications for a lot of different things, which in the scope of this talk, we don't have time to get into. But in the context of art, what it means is that you have that the silhouette of something has to be powerful, that a person's gut reaction upon first seeing something, you have the capacity to sort of tap into this very powerful emotional response. And so, in my opinion, and one thing I really strive to do in all of my pieces is to have that initial gut reaction, that silhouette be strong to take advantage of this reality of the way that our brains work. I'll quickly show you a couple other pieces I've been working on in the meantime since this piece. This is a piece called Action Potential, which is also a microetching. Action potential, of course, is the way that each one of our neurons fires to communicate with the other ones downstream. And what you're looking at here is there's many, many small neurons which are sending their own action potentials onto the dendrites of the very large one. And that information is summing over the length of the dendrites towards the soma. And if you get an integration of a sufficient quantity of these depolarizing signals, then this large neuron will fire its own action potential. I need a drink. This, again, just to show you that this is a physical object. Here's another one that I just finished. This is not the microetching itself. This is the simulation of what the animation would look like. Whoops. This one's called Brain-Machine Interface, which is a hopeful message about the future of human brains and how they're going to be integrated with electronic devices. And the point of this piece, I'll play it a couple times here. The point of this piece, I think, is really to show, as the expression changes over this, at this moment, kind of encapsulating the natural fear that I think we all feel about the future. You know, integrating our brains with various devices, which is going to happen. And the potential for positive change, I think, is tremendous here. So as the piece evolves, the expression changes as well upon the realization of the profound good that can come from this type of intervention. I mean, the most dramatic, if you haven't seen them, I recommend that you look up neurostimulators for Parkinson's disease, where they insert them into the basal ganglia, and to see a person who is tremoring and basically, unable to move intentionally, to leap up out of their chair upon activation of this neurostimulation device. It's quite profound. So hopefully things go well in this regard. This is a still of the image, or of the etching. Let me see how much time I have here. Yeah, I'll just say one quick thing about how art, things that art has really taught me about the brain and about its construction, because essentially building a brain really helps you to understand it in a lot more depth than I would have understood otherwise. This is a diffusion tensor image of the white matter, and is meant to illustrate what's happening in early development. This is kind of a realization I had about how cool the brain is during that period. You have billions and billions of axons which are growing towards their destinations at this point in development. There is a ton of stuff going on. They're all interacting with one another. And even if you had identical brains, genetically identical, epigenetically identical, the environment is precisely the same, that simply quantum events on the scale of something this large and complex would start to play a dramatic role, such that an axon growing from here to here is going to be controlled by some degree of randomness and is not going to arrive at its precise destination every single time. So there's messiness in the wiring of the brain early on, as a direct result of the fact that it's very difficult to wire correctly, which is one of the reasons that your brain has been designed to accept that degree of randomness, that the brain overconnects early on in development, and then through the way that you use it, you optimize and prune away what it is that you do and don't need. It's a really interesting way that it's kind of overcome this inevitable set of errors that you're going to get in early development. Okay, so I'm going to wrap up a little bit here and just talk about, in my experience, what I've found to be the best sort of formula for creating images, or really not just images, just an idea that has an impact in the world. In this case, in the context of an art and science image, is that the scientific content obviously has to be solid. It's got to be there. It's got to be emotionally engaging. Hopefully, you can create some sort of an aesthetic or style which is distinctive, and which sets it apart from other images that folks may have seen. And one of the tools that I use to be able to achieve that is in kind of seeing what has come before and using that as a negative constraint to work against. So, like, my brain works well when I have too many constraints rather than when I have not enough. So I prefer to work against constraints. And when all those things are in place, that's when you have a moment like this, which doesn't always happen, but when it does, it's gratifying. All right, so these are some of the places where you can see or read about the artwork, and I want to give a huge thanks to everybody who contributed to this project, and obviously to Kathy and to everybody within the faculty who invited me out. Thank you. It's great to be here. And for those who are interested, I've got a website that's got a whole bunch of images and videos on it. Please, for academic purposes, always use my images for free, no problem. Just attribute the images as you normally would. I'm very happy when they're used in an academic context. I've got a mailing list if you would like to sign up down here. I've got a sample of microetching right here if you want to take a look at it. It's coated in glass, so don't worry about damaging it. The lighting in here is not really optimal, but you all have flashlights in your pockets pretty much, so take out your flashlight, turn it on, and then hold it up like this and straight the flashlight across, and you can take a look at it here. Yeah, so thanks all so much for your attention. Oh, yeah, questions. Thank you.
Any questions?
That's a good question. So the question was, do we use a laser to etch the surface? We do not use a laser. We use the combination of the transparencies and a general light source. The UV source just looks like a big bulb, allows us to do the etching all in parallel. So you can make millions and millions of these in a small amount of time, as opposed to with a laser where you'd have to raster over the surface, would take much longer time to do. Although I will say that when you are making a hologram, for example, or basically anything which has got a higher fidelity of etch than what we're making, that's exactly what they do. They use a rastering laser over a photoresist to get it.
Any other questions?
Yeah.
Have you done any comparative work with lesser organisms or invertebrates?
Well, the dirty secret is that there's lesser organisms data in this human brain. When we when we couldn't find data for specifically for human brains, we definitely, there's some primates, of course. There's cat, I think, in some of the anatomy. And yeah, I mean, I grew up in a lab doing mouse research, flies as well. So I'm influenced by that stuff as well. And I have other pieces that are along those lines. Yeah.
So, are you doing any current work where you look at certain pathways or certain interactions, or what are you currently working?
Right now, I'm embarking on a project about neuropsychiatric and neurodegenerative disorders. So this is basically the brain in its fully functional state, or a rough facsimile of that. And I wanted to do a series of pieces which are showing how it can go wrong, essentially. And I'm still trying to figure out. It's a delicate project, and one that I want to make sure I do a wise job on. I think I want to be interviewing patients and clinicians and researchers. And be giving a voice to people who suffer for them, and use it as a public education tool, and hopefully to tour it around the country. So that's kind of what I'm working on at the moment.
Any other questions?
Yeah.
What do you think are some of the ways we can encourage other individuals or other students in the science field to integrate art into something you see here that not only our own research but society as a whole?
The good news is that the question was, how can we encourage others to use art kind of in their presentation of neuroscience to the general public? The good news about neuroscience is that the brain is at the fundamental root of absolutely everything that we do, so it applies to literally all of the arts. So I don't think that it is a good message to say that visual art is always the way that it needs to be presented. I think the best thing for a person to do is to look within themselves, figure out what their own passions are. If they have any kind of penchant for art of any kind, be it writing or dance or music, that they figure out how that interest can overlap with an interesting neuroscientific point, and to make science art based upon that, if that's where their interest lies. 3, 2, 1, done. All right, thank you.